Hi, welcome to this video on Bradford Assay for Quantification of Proteins. This method was published by Bradford in 1976 in a journal called Analytical Biochemistry. It is a highly sensitive and rapid method for the estimation of protein microgram quantity. I will briefly explain the underlying principles, components, and conditions of the assay. In addition, how to set up assay and prepare a standard curve using BSA will also be discussed. Before coming to the underlying principles, I would like to explain the features of the dye which is used in this assay. The dye is called as Kumasi Brilliant Blue G250. Kumasi has been taken from the city where it was extensively used in textile industry. The city name is Kumasi. It is in Ghana, and the G stand here for greenish. So dye is not pure blue or red; it is greenish red or blue color. And 250 was taken as the purity index of purity. So Kumasi Brilliant Blue G 250 dye is used. It exists in three forms. The first one is cationic form, depending on the pH. Cationic form is reddish, greenish reddish, and the second one is anionic form. It gives pure blue color, and it is the bound form. When dye binds with the protein, it becomes bluish color. It is anionic form. Third one is neutral form, which is green in color. It is form at the neutral pH. So basically, the spectral properties of these dyes, these forms are also different. For example, cationic form absorb maximally at 470, whereas anionic form at 595 and neutral form at 650 nanometer. Regarding the underlying principles, it involves the conversion of cationic form of dye into anionic form in presence of protein molecules in the acidic medium. So cationic form is reddish, it may absorb maximally at 470 nanometer. Anionic form is bound form, it is the blue color and it absorbs maximally at 595 nanometer. This is also very important to understand the type of interaction between the dye and the protein molecules that convert a cationic form into anionic form. So I will discuss the type of interaction that are responsible for the conversion of cationic form of the dye into anionic form. So this is protein molecules I have shown here: aspartate, tyrosine, and lysine residues. The the dye preferably interact with lysine, which is positively charged molecule through in electrostatic interaction, and it can also interact with aspartate also through electrostatic interactions. The dye can also interact through van der Waals forces with the backbone of the protein molecule with a hydrophobic amino acids such as tyrosine and tryptophan. It interacts through hydrophobic interactions. So these three types of interactions are responsible for the conversion of cationic form into stable anionic form, which gives blue color. So in summary, the cationic form, which is unstable and which absorb maximally at 470 nanometer, is converted into stable form upon binding with protein in acidic medium. This stable form is anionic form. It absorbs maximally at 595 nanometer, and the color intensity is directly proportional to the amount of protein in the sample. So this is quantitative method. So composition of the working solution of Bradford reagent is as follows. Kumasi G250 is 0.01%. It is dissolved in ethanol finely and then it is filtered. Ethanol is 5% volume by volume final concentration. Phosphoric acid maintain pH around 1 to 1.5. At the final concentration 8.5. So ethanol help to dissolve the Kumasi, whereas phosphoric acid stabilizes the ketonic and the cationic form of the dye. However, it has been experienced that preparation of the Bradford reagent is time consuming and and requires careful handling of the chemicals. Therefore, it is advisable to use the commercially available kits ready to use, following the instructions from the supplier. So to find out the concentration of protein in your sample, set up the assay. Label 6 tubes for BSA as to prepare a standard curve, 1 tube for blank as B, and label 1 tube as S for sample. So add BSA in the amount shown here in microgram, 1, 2, 3, 4, 5, and 6 microgram, nothing in B and S tubes. Then add sample, 2 microliter of sample in S tube, nothing in the remaining tubes. Now add 1 ml of Bradford reagent and mix and incubate for 5 minutes at room temperature. You will see color change in the tubes. You transfer the content into a cuvette and read optical density at 595 nanometer using a colorimeter with a blank which you have prepared here. Now, reusing these optical densities, prepare a curve between the optical densities and the concentration of BSA to find out or to calculate the concentration of protein in your sample. So to calculate the amount of protein in your sample, you prepare first the standard curve between the optical density and the concentration of BSA, and this is straight line. And let's say in your sample the optical density is 1.2. You can calibrate the curve using this value, and you can calculate the amount of protein in your sample. In this case, it is 4.4 microgram per ml. So the method is rapid as the color develop within 2 minutes, and it is stable also as it can last up to 1 hour. So it provides flexibility in operation. It is highly sensitive technique which detect protein in microgram quantity and allow us to do assay in microtiter plate as well. Except for some detergents such as SDS and Triton X100, there is minimal interference in this method. There are certain precautions which must be observed while doing estimation. You must make sure that there is no precipitation of the dye in the Bradford reagent. Prepared fresh reagent or use commercial preparation following the instructions from the supplier. Run assay preferably in duplicate or triplicate to reduce or minimize the standard errors. Always set up assay for sample together with blank and BSA for standard curve preparation simultaneously. In the end, I would like to thank you for watching this video, and I believe that this will be helpful for you to understand the concept behind estimation of protein in a biological or in any sample. And if you think so, kindly like it, share it, and subscribe it. Until next time, take care. Bye bye.